Okay, well, we'll get started. So happy GIS Day, everyone. And in celebration of that, happy GIS Week. So my name's Kelly, and on behalf of the Geospatial Graduate Student Organization, I am happy to introduce some of the CGA fellows who will be giving lightning talks today. And first up, we have Dr. Chris Jones, who is a senior research scholar here in CGA. So with that, let's take it away.
Thanks, Kelly. So today I'm going to be talking about some of the research going on in our lab focused on iterative forecasting and how it helps improve our understanding of ecological processes and management of invasive species, specifically invasive pests. Invasive pests are a global threat costing billions of dollars annually for management and the damages that they cause, and the rate at which they are being introduced continues to increase. This puts increased pressure on decision makers to do more with less resources because they're spreading it out over a larger pool of species. So here we think iterative ecological forecasting has a role to play in that. So iterative ecological forecasting is taking theory and data and creating a model of your system and then building a forecast and continually doing this over and over again. And we've been making models for decades in ecology, but rarely do we do it iteratively and continue updating over time, largely because grants don't go on cycles that allow you to have a nice long continuous data stream. But they've been doing this for weather forecasting for decades, and it's dramatically improved forecasts. So there's a new movement in ecology to really create these near-term iterative ecological forecasts, and our lab has been doing that for the past 6 years. Using this POPs forecasting platform, where what we're doing is we're taking these field observations and scientific discoveries that come from the lab and the field, applying them to our models, and we've shown that that process alone has increased our model performance dramatically, particularly for spotted lanternfly. Multiple pieces of data that have come from the lab, including temperature sensitivity for the species and how they've interacted with anthropogenic sources like railways. And then we combine all that and use that with our stakeholders to incorporate their knowledge of the local systems and what's going on to help improve management through this POPs dashboard interface, which has two different methods of interaction, a tangible landscape and a dashboard interface. And the engine behind this is the POPs model. It's a spatially and temporally interactive model that on complex landscapes where the heterogeneity comes from temperature and host availability, and as well as these railways. And what we're doing is we're simulating reproduction, dispersal, and establishment across the entire landscape. But what we don't really know for this system is how much the spatial resolutions of these grid cells I'm showing affect our model results. So one of the grad students in our lab, Britt Lagunas, is performing research looking at answering some of these questions. First, can we accurately predict pest spread at finer resolutions? And the common theory in ecology is as you go to finer spatial resolutions and finer spatial details, it gets harder and harder to predict ecological processes. And is it feasible to make these predictions at these finer resolutions? In other words, can we actually produce them in a time frame in which they're meaningful decision makers? And how does increasing the resolution of the pest spread forecast impact management resource allocation in terms of amount of area they would have to survey and manage if they were to use the forecast? So we did this doing a hindcast for 4 years from 2020 to 2023 at 5 different spatial resolutions, 100, 300, 500, 700, and 900 meters across the eastern US using spotted lanternfly as our case study. And what we found is that at these smaller resolutions, we're actually able to accurately still maintain our predictive capacity, except for when the data was relatively sparse, specifically in the 2020 year, where we had 1/10 of the normal amount of data collected due to the COVID-19 pandemic, preventing them from getting out there and surveying. So as we decrease spatial resolution and data becomes more sparse, we are less able to predict these ecological processes. Which is really exciting that we're still able to get this accurate prediction, but we're not able to do it in a timely manner on like available resources. So we run at 100 meters, it would require an HPC because it's taking approximately 400 gigabytes of memory to run these models, whereas we are able to run on a PC at 300. And if we were to run the entire iterative process that I was talking about, it would take 555 days to do it at 100 meters. So that's not a time frame in which decision makers want to act and want to use these forecasts. They want them in a relatively time. Whereas at 300 meters, it only takes a month. So we can actually use 300 meters as our spatial resolution in a time frame in which would be meaningful to decision makers. And so what we're looking at here is we've divided our study area into 3 regions, a regional scale, and then an interior and an exterior. And we're showing 2020 and 2023 forecasts at 3 of the resolutions, 100, 500, and 900 meters. And the probability of SLF being, spotted lanternfly being in a cell. So what we can see is that reducing the spatial resolution of our data dramatically decreases the amount of area in which our decision makers have to survey. So this is really exciting. We're showing that we can provide timely, accurate forecasts and greatly reduce the amount of area they have to survey, making for a more impactful decision support system. And this was one of the key areas that had not been addressed in any of the invasion literature that we'd seen. So one of the other things that we want to look at now is we want to highlight into that sensitivity analysis aspect. And our lab, before we do that, let's define some terms. So initial condition uncertainty for us arises from the initial locations and number of pests being imperfectly measured because they're visual inspections. Driver uncertainty comes from the weather and host data being modeled across the entire landscape from a few observations. Our parameter uncertainty comes from that calibration process I talked about and the uncertainty around those calibrated parameters. And our process error comes from stochasticity in the model, so just pure random number generation. So our lab performed a literature review looking at 1,000 papers on the modeling the spread of invasive species, and only 268 of those discussed uncertainty, and almost none of them, rounding to 0% of the papers, quantified and propagated uncertainty through their model and partitioned it. And when we talk about quantifying and partitioning uncertainty, this is work being done by Eli Horner in our lab. We're looking at that blue window of uncertainty around the prediction and quantifying where and what sources that uncertainty arises from. So looking at host, initial conditions, weather, parameters, and processing of our model. And so if we do this over time, we see that overall initial condition uncertain, or the overall uncertainty increases, and the effects from all of these remain relatively flat. Now, this doesn't match with ecological theory, which suggests that these two kind of take over as you go through time. The parameter and the processes kind of take over as the dominant sources. And what we've noticed is that all of those studies on this ecological theory piece of this are done on a single point in time. So when we look at this, this is 5 years, and this is Curry County, Oregon, which is the southernmost county in Oregon on the coast. We see that initial conditions are driving the initial area near all the outbreaks, while host is driving kind of our leading edge and kind of those long distance outbreaks in terms of their influence on model, which is really interesting because it allows us to target data collection for host availability and initial conditions in the right locations, leading to better decisions for our decision makers. So with that, and coming back to this, what we've realized is that targeting those initial conditions, which we're working on in the lab through satellite remote detection and early detection with our stakeholders and better maps of our host availability, can really reduce those uncertainties in key areas where our stakeholders really care and really want to be able to manage. And with that, I will thank everyone that's been part of this research, our funding agencies and partners, and take any questions that we have. I apologize for the lack of detail. I was trying to fit a lot into a very short talk.
When it comes to monitoring like a spotted lanternfly, I mean, is there such a thing as a like true negative over a really large pixel? I guess just
Over
how
a
do you?
large pixel, no. There we're taking, so typically for that system, we have like 150,000 samples each year that come in. And so if a cell has like 10 negatives, we treat it as a true negative. Just, we know it's not, but right, or it's impossible to know for sure, but yeah. It really is numbers based at that point because
Yeah, I just didn't know if there's like something with error metrics. I don't know. It just seems like a tricky case where.
yeah, it's also one of those things where it's imperfect. The visual surveys are imperfect too, right? Each one. So that's where that uncertainty piece is kind of coming in, is to understand like where and how those play into it.
That multi-scale analysis, that's fascinating. Have you ever considered the idea of doing it in a dynamic way where you take the coarser scales to identify key hotspots and then dive deep with finer resolution within those coarser region scales? You use 900, but it could even be more than that, right?
Yeah. No, that's a great idea. And we've considered it, and it becomes kind of a computational monstrosity.
Okay.
Like it's already a pretty complex model, and then like adding that like extra layer of dynamic interaction into it. We've yet to come up with an elegant way to do it. But we have thought about it many times. Courtney.
So have you guys considered adding like a management plan within this model too? So like, let's say, okay, there's spotted lanternfly to be predicted here. We're gonna apply pesticides. Are you incorporating those management plans in this?
Yeah, so management actions can be incorporated. In that analysis, they weren't, because we were specifically looking at the effect of scale and not the, but Mark Feinberg in our lab is looking at optimal management and management from different stakeholder perspectives and those types of questions. And we worked with part of that like interaction piece that we're working with stakeholders is they apply the management and see how it changes the outcomes, and then they actually change how they manage based on some of.
We have time for like one more question. There is one. And assume he's monitoring the Zoom as well.
If I were to want to like use POPs, like where could I find it? Is it open source or proprietary?
Yeah, it's open source, so it's completely the, normally I have a download link on here. I think I just forgot to change the text to black, so you can't see it. So right here, there's usually like hey, where to get it. So yeah, good call, yes.
What's next?
We've been focusing a lot on early detection and optimal management, and some of the stuff that Tom back there is working on in terms of mapping hosts via Google Street View imagery and then connecting that to satellite data. So lots of like increasing the data, since when we now have shown that we can do this at smaller spatial resolutions. We need data to support those smaller spatial resolutions.
Let's give it up for Dr. Christian. And next up, we have Dr. Brian Rich, and he is the Gertrude M. Cox Distinguished Professor in the Department of Statistics.
Thanks for coming, everyone. Good to see you all here. I assume you're here for a desire to learn about advanced statistical methods and not the free pizza. Okay, so I'm in the Department of Statistics, so I think I'm kind of an outlier in the room. So I'm a fairly applied statistician, but I still spend about half my time just thinking about equations and proofs and algorithms and so forth. But I spend the other half of my time working on problems, and a lot of the problems that I work on are geospatial type problems. So I work a lot in spatial statistics, causal inference, so a few other places like this. So what I'm going to do is just go through a few projects that I'm working on. Some of the time I'll talk about the cool algorithms and stuff that we have. Some of the times I'll talk about the applications. I hope something sparks a collaboration with some of you. So I'll go through a few just opportunities, maybe for just general collaborations before diving into the specifics of my work. Great. So first I want to say we have a training grant. I'm the PI and somebody from Applied Math, this person, Meta, is the co-PI. We have a 5-year project. It's called Uncertainty Quantification for Life Sciences. Uncertainty quantifications is what Chris just talked about, thinking about all these different sources of uncertainty and blending statistical models with mathematical models or biology models. So we have a huge group of people. We meet every Friday. We have working groups. We have a lot of problems, but definitely we would love to have more collaborations in here. So if you have a really cool problem like the one that Chris just described and you want some extra statistical help on this, we would love to work with you. We have tons of students looking for cool problems. So this would be a great opportunity for collaboration. Another program that I run is the DRUMS, the Directed Research in Undergrad Math and Statistics. This is a summer program for undergraduate students. So every year we recruit 20 grad students from across the country, and they come and they work for 10 weeks on a problem. Last summer, I had a group of students work with Josh Gray on actually an analysis that I'll show you about before, where we had some remote sensing data and they did some causal inference. So again, we're always looking for collaborations. There's a lot of really great talent in this undergrad pool. Like, we get really good students. We get like the best students in the country at this undergrad project, and they're dedicated for 10 weeks. So if there's a nice project that has a cool statistical application, we'd love to work with you on that too. Okay, so let me go into some of my research areas. I'm going to skim over this stuff at even a higher level maybe than Chris did. So one thing I've been working on a lot is spatial causal inference. Causal inference is one of the hottest areas in statistics. The classic line is correlation does not imply causation. But we don't want to stop there. We want to do a little bit better than that. We want to get as close to causation as we want, because nobody really cares about correlation. You care about causation, right? So a lot of problems you can't get at causation unless you have a randomized controlled trial or something. And in spatial problems, what we're dealing with, you're never going to, well, not never, but very rarely are you going to be able to randomly assign treatment groups and do a controlled trial that way. So here's an example. This is what my undergrad students worked with Josh on. The treatment variable is concessions, no concessions. This is the government giving land to private farmers, and then the outcome is deforestation. So obviously you can't just randomly throw plots of land to sell. So there maybe is some bias in the way that these treatments are allocated. So causal inference is a huge field. Propensity scores, matching, all this stuff is what people do. But I work specifically on spatial causal inference because these are the sorts of problems that I often run into. So some challenges specific for spatial dependence, or for spatial analysis, are the correlation between observations. So most classical causal inference stuff, you try to get down to a controlled trial where you're randomly assigning patients individual treatments. Whereas here, it's not clear that makes sense because you have pixels or spatial locations that are correlated, so you can't really think about randomly assigning subjects. It's this whole joint distribution. So just rethinking all the definitions when you have the spatial dependence is really interesting to us. And then there's also this issue of spillover, which is if you treat one location, it doesn't necessarily only affect the outcome at that location, it might affect nearby locations. So in this case, I don't know, maybe next to a place where the land has been conceded to private farmers, maybe that has some impact on the neighboring pixels. So this is another challenge that we're kicking around. So the technical approach that we've been using, we have a few of these, I suppose, but the one that I probably like the best is double machine learning. This comes from the AI ML kind of community where machine learning for them is like a random forest or a deep learning. Our machine learning algorithm is going to be creaking, which is not exactly the state of the art, ML algorithm. It's 100 years old or 50 years old. But I think it's a really neat application for this. And so this is really simple. So, I mean, these equations, like I can just describe the whole procedure here. So you want to do a spatial causal inference. You want to know if X causes Y. One issue you might account for is there could be a missing confounder. So this is a variable that is correlated with both X and Y. Okay, so if you just ignore that, you just do regular regression, you could get biased estimates. Okay, so all we do to remove this thing, we do cross-fitting, which means it's like cross-validation. You just split the data into 10 groups or something. Then you use 9 to build a model and you predict on the last 10. You repeat this all the way through, and then you get predictions for all the locations. And then all you do is you take the residuals from those predictions and you do least squares. So super simple. It's the algorithm you were going to use anyways, except you do it a bunch of times. And then we go through and we have this like 10-page proof that under certain conditions, this works. So the conditions to me are the interesting part because when you're talking about this correlation and causation, how are they related? You can never really get at causation unless you do a randomized clinical trial. So what you can get from a statistical work like this is the things that need to be true for you to interpret your result as causal inference. So in this case, the assumption that you have to make is this missing confounder is a smooth spatial process. So like the number of derivatives is bigger than 2 in our case. And so it's unverifiable at least, but at least it gives you some, like instead of just saying, well, we can't do anything, you can say, well, we can't assume it's causation if this assumption is met. All right, another general area that I work on is extreme value analysis. As you all know, extremes are the most interesting part of a lot of data sets, especially in climate change. Typical statistical analysis, you make a histogram of the data. The big ones that look awkward, you throw those away as outliers and you focus on the middle of the distribution. In extreme value analysis, you take the data set, you throw away the stuff in the middle, which is not interesting because it's not extreme, and then you focus on these 10 observations that are really big. And so super interesting problems from a statistical perspective because we're no longer talking about means and variances. That's the bulk of the distribution. That's day-to-day stuff we're not interested in. What we care about are the extremes. So we have different distributions, different measures of dependence, different parametric models. Really interesting is how you do spatial modeling of these things. So we don't want to assume normal distributions and correlations. That's sort of the foundation of spatial statistics. So there's whole new sets of processes that people have developed to capture this extremal dependence, which is like the probability that 2 sites close to each other are both simultaneously experience a 20-year event. Okay, so working out the theory for this and the computation has been great. It's been super fun, so we've been working on it for a while. This is a collaboration with Sankar from Civil Engineering, who's also a CGA person. The latest thing we're working on in this line of work is studying PFAS source apportionment. So this is a collaboration with people at the EPA. This is the state of California, probably got that one. And the black dots are wells. So a lot of people outside of cities use well water. Sometimes these are contaminated with PFAS, these are the forever chemicals that are recently been regulated by the EPA. Okay, so a lot of these have PFAS on the right as a histogram of one kind of PFAS. And you can see it's like if you're looking at these things, I was talking about the bulk of the distribution and the extremes. This is what I mean. Like normally you'd want to do a regression, you'd say these are too big, throw them out and focus on this. But if you want to understand PFAS and who's exposed, these are the things that are really interesting. Okay. So in this particular example, we also have from a different data source, the locations of all the potential sources. So sources would be like, where are the airports, where are the chemical manufacturing plants, where are the national defense locations and so forth. And so our goal is to try to figure out, ideally, what proportion of PFAS in California well water can be attributed to airports. And one other thing I wanted to mention is sort of fundamental computation work on spatial statistics. So 10, 20 years ago, when I was a lad, if we had 1,000 spatial locations, that was a big data set. That would be like, you can do it, but it's going to take a couple hours on a dedicated machine. Here is a data that we analyzed recently. This is 3 million remotely sensed AOD measurements. This is over space and time. So 3 million observations 20 years ago, we probably couldn't have even loaded in our computer. Definitely could not have done a full spatial analysis of this. Even more complicated is if you're talking about huge data sets like this, the usual simple methods are obviously wrong. So what we normally assume is stationarity, which means that the distribution of AOD is the same no matter where you are. It has the same mean, variance, correlation, and so forth. Talking about the whole globe, that means you have the same distribution over the ocean, in the deserts, in a rainforest. That's ridiculous, right? So we want a fancier model that allows for these different distributions in different parts of the world. So we're fitting a huge data set with a really fancy model. And so we developed this method. It uses, well, I won't bore you with this. The interesting part is you rewrite the spatial model into a way that it happens to look like a deep learning model. So it's not like a typical deep learning model. It's not like a CNN or anything like this. It still is our regular normal distribution model with spatial correlations. It's just, you use this spectral embedding things to approximate it that way. And then we can use all the deep learning software, and that is super fast. So in this analysis, we're able to do the 3 million observations in 39 seconds as a quick analysis and a few minutes or something as our formal analysis. And you can see it's locally adaptive. So the width of the prediction intervals are much higher where there's high AOD, low AOD. So for me, this is pretty remarkable. So, deep learning is probably going to ruin the world and the robots are going to take over. But at least in the medium term, exploiting these computational algorithms helps me just fit the models that I wanted to fit before, just in like a millionth of the time. So this has been really exciting, thinking about how to not like replace our statistical models with deep learning, but how to use those tools to help us improve our computation. Okay, I think that's all I wanted to say. Thanks, guys.
Thank you very much for your great talk and outlining lots of examples. I'm curious, what do you use in terms of computational resources for this one in particular? And then if you could go back to the first slide, the first opportunity on quantifying uncertainties, how can students join? Like, what do they need to have good grades? Do they need, like, what do they need to join?
Okay, on your first question, my student did a summer internship at a national lab. So he used like all the Python deep learning stuff, but the key was it was at a national lab with a gazillion GPUs, and like, this is not a laptop 39 seconds. It's a serious computer.
Google that type of thing.
Yeah.
Okay, cool.
Okay, so for this thing, we have our fellows that are funded by the grant, but I think probably those are all going to be math and stat PhD students. So that's how it's set up, and we have a couple of postdocs too. But we also have a whole bunch of people that just come to our meetings and listen to our talks and engage in working groups and so forth. So I'd say that's open to pretty much anyone, as long as you're interested in like coming regularly and meeting with us. And especially if you have a cool problem and cool data, we'd love to hang out with you. Any questions?
For the spatial causation analysis, so in your model of double machine learning, like Z was, of course it was simplified, so there's only Z. But I'm sure like the bias would be very like non-uniformly like existing in different space. Like what is simplified in this?
Function.
Well, I mean, our whole approach is very simplified in that we assume the degree of bias is the same everywhere. We also basically just assume the same model applies everywhere. So thinking about how to apply this locally would be a really nice approach.
Well, what is application actually, like, what kind of causation were you looking for?
This is one example. So looking at how do these treatments affect deforestation?
Deforestation.
We've looked at other things too, like how does air pollution affect health and a bunch of other stuff like this.
I would love to know more about the applications for the extreme research, specifically like the example that you had, the stream flow. What are you taking that?
Well, for streamflow, we're now trying to incorporate climate models into this.
Okay,
So this is just take the historical data and look at trends in the past to see where streamflow is becoming more or less extreme over time. Another thing we're working on with Sankar is thinking about how to project this forward 20 years or something, different climate runs and account for that sort of uncertainty, but all still embedded in this extreme value analysis.
that's where you're heading, or have you accomplished that?
We have done some very simple analysis of this, but I think the full, like, what would satisfy me is a statistical analysis, I guess.
What does double machine learning means in this case? I think you might have explained, but I don't think I understand.
Yeah. Double machine learning refers to the fact that it's in two stages. So you first do this thing where you make the predictions and kind of remove these trends. And then on the residuals, you do another analysis. So it's machine learning in the first stage, and then the second stage is called machine.
Are the California and example with the PFAS measurements a different definition of extreme observations than the stream information?
Different definition.
Well, it looks like you're looking at extreme, like count of observations and number of PFAS. But all the observations in the other one are extreme flows in different spatial locations. So it's a different model.
Yeah, it's a different approach because I guess, yeah, this is a subtle difference, but in the streamflow example, we took at each station the annual maximum. So all the observations were extreme in that sense. And then we threw out all the daily data, we just kept the unit max. And in this one, we're analyzing all the data.
Right.
Because here we don't want to just focus on the extremes, like we want to know what percentage for airports. So that could come from a bunch of small sources or a couple of big sources. So we want to keep them all. So here we're trying to analyze the whole distribution, but the most impactful ones are going to be the big ones here. And so we kind of, this is like a histogram of all the PFAS. So we fit a distribution that kind of looks like this, where it's got some mass that's low, but also a really heavy tail to capture these guys.
Do you have a favorite type of Bayesian method that you like to use?
It's like picking between children.
No, you have a favorite.
Well, because it has a cool name, I will talk about our R2D2 prior. So this is a approach for linear regression. So R squared is like the common measure of model complexity and linear regression. So that's the first R2. And instead of putting priors on like individual model parameters, if you know Bayesian stuff, we put a prior on R squared. So the idea is that this would allow researchers to say, okay, I don't know, like a genetic analysis, maybe you have some outcomes and you have 1 million genes or something. You have no idea what the effects of the genes are, but maybe you know from some studies that R squared should be about 0.1. So you can kind of fix 0.1 as R squared. And then we do variable selection within all the genes to try to pick off subsets that are important. So
Let's give a round of applause. And next up, we have Dr. Natalie Nelson, and she's an associate professor and university faculty scholar in the biological and agricultural engineering part.
All right, thanks everyone. So I took a slightly different approach to this talk. I'm going to be going at a very high level over multiple projects that are all motivated by basically one consistent decision-making activity. And so I'll spend a little bit of time on that decision-making activity up front, and then I'll do this kind of quick blow through several projects. And so I'm the PI of the Coastal and Watershed Analytics Lab. So basically what our research team does is characterize and model non-point source pollution. So this would be pollution that occurs in a diffuse way across the landscape. And the main types of pollutants that we focus on are ones that affect surface waters. So we look at nutrients and then how that can ultimately lead to downstream consequences like harmful algal blooms. We also look at fecal pollution. So basically how does feces end up in the water and lead to issues with contamination, whether it's of shellfish or then leading to issues like needing to post swimming advisories. So we try to understand where this is happening and why it's happening. And we use a range of methods that vary from like basic kind of data science methods, process-based models. And then we also do some field work, especially in cases where existing environmental monitoring data sets are insufficient to be able to address these types of questions. All right, so I said that I would present basically one piece of background on this motivating activity, and then all the projects are going to relate to that. And so what I'll be talking about is issues of fecal contamination in shellfish growing areas. And it's important to sort of understand as background that stormwater runoff is the primary driver of fecal bacteria concentrations in coastal waters. So basically right after it rains, you have a flushing of the land surface, and that can transport feces from land into coastal waters. But you also have issues of septic system failures and wastewater treatment plant failures immediately after storms, because there's basically just increased load from all the water. And so this can be a problem, especially when it comes to shellfish, because they filter feed. So they take up particulates around them, and that means that they can also ingest incidentally some fecal bacteria, which actually isn't a big deal for the shellfish, but is a big deal for you if you consume shellfish that's been harvested in contaminated waters. So especially in the case of like raw oysters, you could get sick if they were harvested in contaminated waters. And so there are federal and state regulations in place to prevent the harvest of shellfish from contaminated waters. And so basically what you need to know is that coastal waters, so here we're looking at part of the North Carolina coast, they're divided into these administrative units called shellfish growing areas. You can see that they're just sort of straight lined polygons. And so these polygons then will be assigned different management criteria where basically the state regulatory authority can say this area is temporarily closed because we're worried the water quality is bad. And so in North Carolina, the system that's used is rainfall thresholds. So these are basically rainfall depths that can fall within a 24-hour period that would lead to a management action. So here, the darkest red, this is corresponding to a 1-inch rainfall threshold. This is the Newport River estuary. So basically, if there's 1 inch of rainfall in 24 hours, they immediately will close that area to harvest to ensure that there aren't any shellfish contamination issues. And so the decision-making activity then, this management process, is that rain will exceed the 24-hour threshold. The shellfish growing area is temporarily closed to harvest, so it's a conservative management action. The regulators are going to wait until they think the waters have returned to baseline or safe conditions, and then they're going to go out and collect a sample. They're going to measure fecal bacteria from that sample. It's going to take at least 24 hours because there's an incubation step as part of that analysis. And so a day later, they will then have data from which they can decide if the waterway can be reopened or if the closure needs to continue. So this is a management process that is ultimately protective of public health, which is important, but it is really inefficient. So there are a lot of opportunities to improve this process through the use of different types of technologies and models. And that's a lot of what has been motivating different projects in my lab. And so as one example here, one of my former students who I co-advised with Angela Harris in the civil engineering department was Julia Harrison. And so Julia had been testing out basically different types of sensing platforms that we could deploy in situ to help us then detect in more real time when these types of fecal contamination events could be occurring. So you can't measure fecal bacteria directly, but you can measure several proxies of fecal bacteria in real time.
So she was testing out some novel sensing systems as well as then developing nowcast models, with the idea being that you could have a nowcast model that could issue real-time estimates of fecal contamination, such that either you as a person of the public could decide if you wanted to go out and swim. And then from a shellfish perspective, it helps them to understand when they could go out and sample to assess if waters have returned to baseline. These shellfish sanitation programs, the state regulatory authorities are also collecting a lot of monitoring data so they can understand if a waterway is changing in terms of its baseline conditions. And so we use that historical data to help us develop different types of models. So my former student, Nat Chazelle, had she had done an analysis of the North Carolina data and then also used the data that were collected in Florida across all of their shellfish growing areas along the state to develop forecast models. So here she was using weather forecasts from the National Weather Service and then taking other information on landscape characteristics like land use, soil drainage class, and using that to then predict fecal bacteria concentrations today, tomorrow, and in 2 days. So this type of forecast model is mostly geared towards regulators to help them anticipate how they might need to allocate resources to help them with then managing these different closures that can happen over really large areas. We've also created a decision support tool called Shellcast. So this is still in operation. It was created a few years ago. This was initially led by Sheila Saya, who was a postdoc at the time, and is now being maintained by Makiko Shukunove, who's in CGA. And so with Shellcast, we take forecast data from the National Weather Service. We use their probabilistic quantitative precipitation forecast, and we couple that information with the management thresholds that are used to then issue predictions every day about whether or not an area is going to close, but we do it just based on rainfall. We don't account for historical water quality information. So here we're using the decision-making tool. We're ensuring that it matches with the same types of information that regulators are using to make these decisions. And so through the tool, we'll have predictions about which areas might close today, tomorrow, or in 2 days. And there's also additional functionality like having text and email notifications. And so through our work, but also the work of several others, we know that salinity is the best predictor of these fecal contamination events in places where stormwater runoff is really driving the fecal contamination. So you have a storm that occurs, there's more fresh water that's coming into the estuary, salinity drops, and so salinity ends up being a pretty good proxy. So my former student, Julia, I mentioned she deployed sophisticated sensing systems with a full suite of water quality sensors, and they're really expensive. What we found is we can probably in many cases get away with just salinity data, but there are very few places where we have salinity monitoring data that are collected continuously and in real time. So we've been exploring ways that we could partner with other types of groups to collect this data and not just rely on, for example, government offices that are already pretty resource lean. So we've been exploring ways to partner with oyster farmers, and we created a pilot program called Sea Salt, through which we had deployed sensors at their farms, and we worked through the mechanics of understanding how you could do this type of community-based monitoring. So we also worked with Dr. Whitney Nolenberg to do interviews and collect more qualitative information as well to help us with expanding this program in the future. So hopefully, if we can find a suitable funding opportunity, we can expand this. And this would help us with the fecal contamination predictions, but salinity data are used across many different applications, so this would benefit many different studies. And then finally, we've been thinking a lot about connections between rain and stormwater runoff and contamination events in estuaries. But we've also been trying to think about, well, what are other drivers aside from rain that could be contributing to fecal contamination events? And so one phenomenon in particular that we've been studying is tidal flooding, which is occurring at increasing frequencies in coastal communities because of sea level rise. So in many places, there hadn't been much flooding at all. And then in more recent years, they're starting to have these chronic flood occurrences as a function of tidal variation. And so my former student, Megan Carr, had done some of our initial work to characterize the water quality of these floods, specifically with regards to fecal bacteria. And now Kelly, and then also another student, Jenna, who I co-advise again with Angela Harris, is helping to expand that work by accounting for factors like septic tank locations and understanding how different types of land use can result in different flood water quality patterns. And we'll also very soon be starting to connect what we see in the floods to also what's happening in estuarine waters. So that's the next step. And that's it for me. So thanks for your time.
Great talk. I like the sea salt like name. I thought it was really interesting. But so this seems like a really great tool for managers, but also the public. So it looks like you have a lot of dashboards. For someone that eats like shellfish a lot, this might be like a great resource to look at. Are you opening these up to the public as well?
Yeah, so for the Shellcast tool, that's already available. I didn't have a link, but that's just a website that anyone can go to. For sea salt, one of the goals of the project was also to understand comfort with data sharing. And what we found is that there is definitely some concern with sharing the data fully publicly. So there was a lot of openness to share the data with researchers, to share the data with peers, and then the public was where there was more resistance. And it wasn't because there's anything to hide, it was sort of like, what unintended consequences could there be to us disclosing this data collected from our farms? And I think concern that maybe people would see, oh, the salinity dropped, why didn't they close this area or something like that? So we need to explore some quasi-anonymization options. So for example, could we say these data were collected in this segment of the estuary without disclosing the coordinates? But we didn't get there with this project. So I couldn't tell you yet what is the level of comfort they have. But otherwise, the data that I present, or the projects, the data are all publicly available.
Lovely, thank you. That was a great talk. So I have a couple of questions. One is about relating the floods with the water quality. So I was wondering if flash floods in particular would be important to map and link to water quality. And then a somewhat related question is, I know that in your work, you also looked at swine CAFOs. And I was wondering, I mean, there's clearly a direct link with the manure lagoons, but would poultry CAFOs also impact the water quality? And how would you be able to make that link, I guess, because it's not as clear as there is water from the manure lagoon that goes into whatever stream and impacts the water quality. And then lastly, the Shellcast, what was the software that was used for that?
Yeah, okay. All right. Question one was, don't even remember.
Flash floods.
Flash flooding. Okay, so flash floods. Yeah, I mean, flash floods, I haven't really gone into that world much. Like I was part of a project that looked at flooding following Hurricane Florence. But these, in a way, they have some characteristics of flash floods, not the speed, but they're occurring chronically. And Catherine Anardi is the PI of a NASA grant that is seeking to do some mapping via satellite remote sensing of the floods because they're really poorly documented. So that would be very helpful to have that information, especially like the spatial information of flood extent would be very useful for being able to then relate what we see in the estuary. The second question you asked was with regards to the lagoons. So my understanding is that most of the bacteria that you see in coastal systems originates from pretty short distances. So it's really what's happening immediately at the coast. And so for the most part, from what I haven't done this research myself, but I don't think that there are indications that the CAFOs that you have mostly then further up into the coastal plain are affecting this kind of flashy water quality because it tends to be pretty short-lived. So it's really immediately after the storms. So it hasn't really been as much of a concern. But then in inland waters, definitely concerned. And your final question.
This software for
Software, yes. So it initially was created with a combination of R and Python and is now, I think Makiko has migrated everything to Python, but I don't feel 100% confident. I had been more involved with the initial development and then Makiko has helped us to expand into other states and to do things in a much more efficient way. But I think it's mostly Python based. We use Google, the Google Cloud platform to deploy.
Thank you so much. It was a great talk. I'm curious, as someone who really likes to eat seafood and specifically raw seafood, like what your thoughts are on the safety of eating raw oysters and how maybe that's changed throughout your research.
Yeah, there are really good systems in place, like they're conservative management practices, right? So in many cases, you don't, like for swimming, you don't see the same type of conservative management practice. So they rely more so on grab sampling data to issue swimming advisories. So they do take a conservative approach because the stakes are higher because people are consuming the shellfish. It is imperfect though. It's an imperfect system. I'm someone who's had food poisoning and stomach viruses 3 times in the past month. So right now my feeling is I'm not eating raw seafood for a long time. But I think there are good systems in place. Like I actually do think that the regulatory system is quite good, but it is imperfect. Yeah. So it depends on your risk tolerance.
Kind of building up like that regulatory component, what goes into determining the threshold, if you know, is it like the size of the watershed or some baseline characteristics? or what kind of
Empirical.
stuff?
So they collect data. They have historical monitoring data that they collect, and then they'll also sometimes run studies, and they basically estimate the sensitivity of the system to rain. But then there are other places. So in North Carolina, it's done 24 hours, then rainfall thresholds, and there is a need to have a certain level of simplicity with the management structure. In Florida, for example, they will use multi-day thresholds, so up to 7 days. And they also use river stage in some places because they find that that is a better proxy as opposed to just rainfall. So if the river stage is of a certain height, then they'll go ahead and close the waterway. But so it's mostly done through empirical analysis, and there's actually, in some cases, they're not allowed to use models like per the regs.
So regarding the sea salt data set, so I think there are some monitoring stations in the past along the coast, but some of them are not functioning. So have you ever considered to ask if there is a way to maybe resume data collection from some of those locations so that people can at least have consistent historical data for some of those locations? Has that been part of the discussion regarding the project or not?
Yeah, that's a great question. I mean, I haven't asked anyone. I guess I would say that, so in terms of thinking about barriers to expanding this type of monitoring, one is capital cost, but that's actually, I think, not a huge barrier. So the initial cost of purchasing equipment. What is a much larger barrier is the ability to maintain the sensors, like what you're describing. So we can often get grants that help us purchase equipment or might allow for that initial deployment and data collection. And it's the long-term maintenance that's really, really challenging. And so I've also heard this from, for example, the Division of Marine Fisheries. They have a lot of really impressive equipment in a locker that they don't deploy because they don't have the resources to go out and maintain the equipment. So that's why we've been interested in this idea of partnering with oyster farmers, because they already are going to the same locations frequently. They have their own motivations for getting the data. So it seems like it could be win-win. I agree that I hadn't thought of it before, but I think then trying to find places where we could match up with historical data that's been discontinued would be really valuable for increasing the data record. But we haven't done that.
And let's give Natalie a round of applause. So thank you all for coming to the TGA Fellow Lightning Talks. And we have a forum tomorrow and also a GRASS GIS workshop to continue our celebration of GIS Week. And for Friday, for CGA students, we also have a trivia event. So thank you to the 3 of you for coming to speak with us today, and hope you have a good week.
With the